and thank you all for having me here today. So, as you might know, I work on transposable elements, mostly in Drosophila, and today I will tell you what I learned during these 3 years of research here at Vetman. So, to start, maybe it's good to remind you all a bit about transposable elements, which are these very weird, we might say, inhabitant of the genome. So these mobile, but sometimes not so mobile sequences that jump around the genome, and this is why people usually refer to them as jumping genes. And what is always been striking for me, at least since the beginning, was that there's like a huge uh, variability in the T content if we compare between species. And here is just a small example of that. As you can see in humans, people usually say there's between 45 and 55% of the genome that's made up of Ts, while in the mouse is smaller, it's around 37, 40%, and in Drosophila around 20. But as we will see, these percentages might change over time, and also depends a bit on the parameters you use to classify them. But yeah, so they make up big parts of the genome in many genomes, and if we want to understand evolution, I think we should also look a bit at them because they have been neglected for a long time. At the beginning, when Barbara McClintock discovered them, she was dismissed for the first year. Nobody believed that there were these elements that were moving around, but after a while, she managed to convince the world, and then people started to study them, but it was very difficult because if you work with genomic data, you know how annoying can be to have repetitive sequences, and these are some of the most repetitive sequences you find in the genomes. But luckily for us, in Drosophila melanogaster, they are abundant, but they are not so many. So once again, it's a very good model organism also to study T, and they have been studied in Drosophila melanogaster for a while, and I will tell you something about it later. And we will not enter into the molecular mechanism of Ts, but just as a quick reminder, usually we divide in class 1 and class 2. Class 1 are the retrotransposons, so the one that go through an RNA intermediate, and then they get reverse transcribed in DNA and insert in another part of the genome, while class 2 DNA transposon are follow a cut and paste mechanism. So they just code for an enzyme usually, they cleave and they mobilize somewhere else. Then there are all sorts of variation, and there are also hyperparasitic elements that are unable to mobilize, but they steal, let's say, the enzyme from functional copies, and this also leads to interesting dynamics. But let's move on. So some examples just to convince you that these in evolution are important, and probably some of the most famous example, even though they didn't know at the time they were caused by T, are the pepper moth industrial melanism, a famous textbook example of adaptation that was caused by T, I think a DNA transposon insertion in a gene. And also the wrinkle seed used by Mendel was later discovered that this phenotype was actually caused by a T insertion. And finally, the most recent one that was described last year in a paper on Nature, where they found that ALU insertion that occurred around 20 million years ago probably led to the tail loss in all the hominoids apes. And the one I presented so far, just example of, let's say, large effect mutation. So you can think about these long stretches of DNA that jump sometimes, and if they insert in the genes, they can break them. Or if they insert next to the gene, sometimes they can increase their transcription. Famous example involves pesticides and insecticides, where LTRs, usually if they insert next to a promoter sequence, they can attract more transcription factor and increase the production of the protein that's the target of the pesticide or the insecticide, and allow for resistance. But we also have more sophisticated mechanism in which Ts were co-opted and they, by providing raw material for evolution, they allow the evolution of very interesting traits. I think one of the most interesting one is the evolution of the VDJ recombination, which is shared by, I think, all vertebrates. And this is what allows to have adaptive immunity. So even though we don't produce all types of proteins to fight pathogens through this mechanism. You can basically um, all the vertebrates rediscover evolution within themselves, where you have this recombination event that allow the formation of antibodies, and then this was driven by this RAG1 and RAG2 that were later discovered to be actually ancient transposons that were repurposed for this role, and another famous one in mammals and in placental mammals in particular is syncytin-1, which is an envelope protein that was repurposed to allow placental formation and fusion. And finally, some example from melanogaster that you might know, and also other drosophilids, are the Het-A, TART, and TAR-E uh, non-LTR retrotransposon that are involved in the maintenance of telomeres in Drosophila and melanogaster, because in Drosophilids, the telomerase enzyme, which is what we have in humans and also in many other animals, was lost, and now the telomere maintenance is orchestrated by these three transposable elements. And another recent discovery is the R2 transposable element, which is again a non-LTR one that jumps in arrays of ribosomal DNA and allows the maintenance of a certain size, because these arrays of ribosomal DNA are repeated stretches, and over time they risk to get degraded, and this is a problem because usually ribosomes are the most abundant proteins in several cell types, and so they need to have a certain length, and this transposon found kind of a niche to jump in and counterbalance this force that tends to reduce through recombination the size of ribosomal DNA over generations. Now the question that you might ask is, where are these Ts coming from? So the classical view is just they have been there in your genome, in all genomes, from the beginning, in very distant ancestor, probably they were acquired, and then they multiplied over time, and there was this arm race between host and transposable elements that they managed to overcome and to still stay, let's say, alive or still mobilized. But in recent year, we saw a shift in this view, and what we saw from initially analysis from different lineages is that some Ts appear to have mobilized from one species to the other. And how can you find this? So an idea is to compare the orthologous genes of this species, in which you will expect to have a certain selection, usually purifying selection, and when you look at transposable elements, your expectation would be to have more variability, because there are no constraints. You would expect a pattern of sort of neutral evolution and to accumulate more mutation. So if, for example, you look at two species that diverged 50 million years ago, and you find transposable element sequences that show a lower divergence, a bit like in the panel C of this figure you can see below here, then the genes you might start to wonder, how is this possible? Why this region, these sequences scattered around the genome have maybe just one or two different nucleotides, where genes that are under selection and are more constrained in their evolution, they show maybe hundreds of SNPs. Well, one explanation can be horizontal transfer, which is this concept in which basically the transposable element gets mobilized and in some way that are still not clear to us, gets delivered to another species. And then you will not expect to see much variability on this insertion, because the event might have happened like a few thousand years ago compared to the diversion of the species that was maybe a million of years ago. And then this pattern is explained. So people did that. They went and they compared species that were very distant, and they looked for putative horizontal transfer events. And we will also look at some of them, but in a different way during the talk of today. To talk about transposable elements in Drosophila, we cannot not mention Kidwell's first experiments. And in these experiments, they were very simple at the time, and we still do them 40 years later. What they did was to collect some strains from the wild and cross them with lab strains that were corrected some years before. And what they saw was a very strange pattern. Now, I will simplify a bit from the original paper, but just to give you the idea. So when they did the cross from the lab strains with the wild strains, and the lab strains were males while the wild were female, the F1 progeny was fertile and didn't show particular phenotypes. While when they did the reverse cross, so wild P males with lab M strains, the progeny show different phenotypes, and the most striking one is probably the gonadal dysgenesis. So basically, they were sterile and they show no ovaries. And this was very weird. This goes against like the classical Mendel's law. And after some analysis, they figure out what was the difference between the M strain and the P strain. And here I'm just mentioning one, but there were multiple strains. What they discovered is there was a transposable element that later was called the P element that was caused these sterility phenotypes and a series of other phenotype like recombination in males. And this was very interesting because it was like the first evidence of a strong phenotypic effect caused by Ts that was no insertion specific. So what I mean is like the P element was in around 20 copies, but they were scattered around the genome, and it was not caused by a specific copy that inserted and knock out, for example, a gene, but was the P element per se that was causing this phenotype. And this was very curious. And after the discovery of the P element, there were still unanswered questions like why one direction of the cross works and the other doesn't. And it took a few years, but then people discovered piRNAs. And basically, piRNAs, without delving into the molecular details, are small RNAs produced from some loci that are generally called piRNA clusters that are rich of transposable elements. So you have this small RNA produced, and this will recognize the transposable element in the genome and avoid mobilization. And here, for example, you have the ping pong size called that you maybe heard of. And this is very interesting, and is also the explains the phenotype that they saw before, because usually piRNA are deposited by the mother, and therefore when you do the other direction of the cross, you don't have the deposition of the piRNAs. So in this case, if the P element is in the male, and therefore it will go, let's say, wild in the progeny ovaries and will cause this sterile, this gonadal dysgenesis phenotype. Finally, I want to give you like a view on how we look now at transposable elements. So if we think about a recent event, so a recent transposable element coming into the species, and I forgot to mention that this was the case of the P element. So the reason why the M strains didn't have the P element was because they were collected before, and the P strains collected 10, 20 years later, or any Drosophila you can find now in the wild will carry this transposable element, is because this transposon comes from another species, Drosophila willistoni, which is very distant from Drosophila melanogaster. They don't hybridize, is a South American endemic species, while melanogaster is originally from Africa, and it just recently colonized all the continents by following the human radiation. So when they entered in contact, Drosophila willistoni, through some vector, probably donated this transposable element that then colonized melanogaster. So all the Drosophila melanogaster that you see now have this P element, while if you went in the 1940s or 1930s and tried to collect Drosophila melanogaster, you would have not seen the P element in the genome. So how we look at transposable element invasion now? The classical model, and it's a bit simplifies, but does its job, is this trap model. So you can see maybe on the top right, okay, I cannot make the end up here. On the top right, basically the idea is that after the horizontal transfer, the transposable element is just in a single fly or in few flies. And these flies obviously reproduce, and the transposable element can mobilize in the germline because it's not silenced yet. And it jumps in a new location, and then it keeps jumping until eventually ends up in one of these traps, which are simply region of the genome that are able to produce piRNAs. And after that, this silencing also spreads, and you expect the transposable element to not increase in copy number anymore. And here, the three images you see with different transposition rates are from a paper from Robert that basically show how we imagine T invasion now. And on the x-axis are generation, and then the copy numbers on the y. And conceptually, you have this first phase where the T thrives and this spreads after the initial jump. Now we're looking at from a genetic point of view, so vertically in the population, you keep mobilizing, and at a certain point you reach a phase where it's silenced in every single fly that in this example is showcased in red. And then what happens after? Well, you can imagine that now the population, every fly has different T insertion, that they are all at low frequency, and then the fixation probability of every insertion, assuming them neutral, would be like 1 over 2n. And how many insertion we have? Well, we have the average in the population at that moment, that in this example is 200, times the number of individual, which is 2n. So 200 times 2n times 1 over 2n. So the expected number stays the same. So here you don't see any change, but actually there are some insertion that gets removed, some insertion that increase in frequency, but the overall number over time, after you have the complete silencing, should stay the same. And this is a bit how we look at transposable elements, and this is actually a very fast process. So later I will show you, but we are not imagining this in evolutionary timescale, but in a more in more short timescales. And this was the state of the art when I arrived here 3 years ago, more or less. So there was some evidence that there were horizontal transfer in Drosophila melanogaster. And this was our current view at the time, but they were seen as kind of oddities. So if you read some papers, there were some estimates that maybe an horizontal transfer event in Drosophila is every 200,000 years or something like that. But it already started to look a bit suspicious because we have the P element. It was the last one documented, but people then, after discovering the P element, they look for more and they figure out that actually some other transposon during the last century arrived from different species. And these were Tyrant, an LTR, the I element, a non-LTR, and then the two DNA transposon, OBO and the P element. So one of the first works that Robert mentioned subverted what we were doing in the lab, kind of was the publication of these ancient or old, let's say, not ancient, genomes of Drosophilids from Sweden, north of Germany, that were collected actually at a previous time. So our reasoning was the following. If uh, we look at all the transposable elements that are present now, and we look at all the transposable elements that were actually present in the 19th century, we should see that in these old samples, these 4 are missing, but maybe there are more that were actually missing that arrive and colonize the genome of Drosophila melanogaster during this period of time. And so that's what we did. So basically, you will see a bunch of these plots. These are just coverage plot or sashimi plot. Basically, we tease. In this case, we don't care about the genomic location. So on the x-axis, you just have the sequence of the T. You map the reads on a library of Ts and single copy genes for the normalization, and you get an estimate. And so what I'm showing here in the top panel are the 3 transposable elements that we found were actually missing in the 19th century samples, plus the 4 I previously mentioned. So these are 412, blood, and opus. If you see, still from museum sample, but collected in 1933, just to make sure there was no degradation that was specific for this tea for some reason due to the museum extraction procedure, you can see that all these 3Ts are present. And if you check any sample collected after 1933, you will see these 3Ts in relatively high copy number. As you can see, they are all above 10 per haploid genome. While if you check in the samples collected in the 19th century, and this is just one example of multiple that we have, you will see that they are missing. For 412 and opus is more clear. For blood, it's a bit more confused, as you can see from the plot, and the reason is that there's an old insertion of a distant relative of blood in the centromere where short reads were still mapping. But basically, they show us that actually T invasions, so events of horizontal transfer from another species into our focal species, appear to be not so rare. And below is just a plot keeping all the samples from the museum and not just one. And they show the same thing. And if you check some other transposable elements like Chircha Invader, and we actually check for all the T's in the library, there are no differences. So only these three appear to be missing in the old samples, but are present nowadays. And then we moved on, and we wonder, like, Drosophila melanogaster is the most studied organism, probably one of the most studied model organisms in the world. So we will be surprised to see novel transposable element invasion that happened after the P element, because after the P element, people had way more resources to study Drosophila melanogaster, and there are way more samples. But surprisingly enough, we managed to find one. And this guy, we called him Spoink. It's a bit of a funny reason why, but it's the name of a Pokemon. And this is, again, an LTR retrotransposon. And here we start to see a pattern. I have to tell you that most of the T's in Drosophila melanogaster are actually LTR, so maybe not so surprising. And except for the characteristic of this T, what is really interesting is that, again, he was missing until basically the 60s. So he invaded after the P element, but for the analysis of this one, we have way more data because we can leverage on all the strains collected over the years. And now we are, I think, around 600 or 700 strain collected over the past 200 years. Obviously, there's a bias on the more recent years, that strains that were collected and sequenced. And again, a coverage plot just to show you an example. If we have a strain from the 60s, you basically see no rates mapping on the consensus of Spoink. While if we look at recent strain, we can see there are on average like 30 copies per haploid genome. But since we were lucky with this one, it was a more recent invader, we still have some of the strain that were collected in the past, like in the 40s. And these are basically in a time capsule. So you have the strain that's collected in 40, is maintained in the lab, and if you don't cross it with flies from outside, which is what you usually do if you want to maintain your strain, this strain will be lacking the transposable element. And so we can do some experiment on them, and we can also sequence them again, and maybe with long reads, like in this case. And what we see for long reads analysis is the same. So I will explain you briefly this figure. Again, we have the two strains, one from the 20s and one from 2003. And then we do basically repeat masker on the long read assembly. And the dots you see, if they are red, they are very, very short. So they are less than 25% of the consensus sequence. So they're like less than 1/4 of the entire length of the T. And they are on the y-axis, you see the divergence. So there are some T insertion that maybe have something in common with Spoink that you can see also in the strain from the 30s. But the full length insertions are the blue one with 0% divergence that you see from the example of RAL 737, and also all the other strain we check that basically were collected after the 70s. And we wanted to be sure. We also did some old school PCR on this strain because they are available. We have them in the lab, and we saw the same result. And a curious thing is this transposon is not present in ISO1. ISO1 is a strain that's actually a cross of different strain and comes from the 70s and lacks the T. And why I think this is interesting is because many analyses or many gene annotation and also reference genome often take in account ISO1, and they give for granted, it was, at least in the past, was given for granted that why bother to analyze new strains since horizontal transfer are rare, probably they happen every 1,000 years or something. It cannot be that a strain collected just 5 years later has a transposon that ISO1 is lacking. But actually, this is what happened. So a few years probably after the strain used for ISO1 were collected, Spoink invaded the population of Drosophila melanogaster, and now it's in all the flies you will find, all the flies you will find around the world. And finally, also this from the long read assembly, we can check the different insertions. And what we would expect, as I told you before, from a recent invader is that all the insertions are at a low population frequency, because you have to imagine that a transposon invades, you have the initial insertion that mobilize, create a new copy, then the old one is kind of inherited, but there are new one forming until there's the silence mechanism kick in at the population level. And so you expect them all to be at a very low frequency. And this is what you see. So except for few outliers, all the that are probably not spawing, since in this case the color are the divergent, so these are more than 25% divergent, so we will consider them a different T. All the insertion we find in the long read assemblies from different Drosophilas collected around the world show different insertion of spawing. So basically what I'm telling you, if you collect a fly in the US, you collect a fly in Europe and another one in China, they maybe will all have around 25 insertion of spoings, but every insertion will be in a different position. And this is actually the pattern we expect for recent invaders. Over time, this will mobilize, some will be fixed, some will be lost if we assume just drift, and over time they will increase in frequency. Because it was the minimum that was available here. So basically every insertion has an insertion frequency of basically 1 over n, because you find this insertion in this, and I don't remember how many assemblies were here, but 8 assemblies, yeah, 8 assemblies, 25 insertion, and many dots. So then we wonder basically where is this spawning coming from? And we were lucky because by the time we were doing this analysis, many Drosophilids genomes were released. I think here it's when we had like around 250. And basically what we did, we used the sequence of spawning to look in other species and check if it was present or not. And what we found was that, yes, it was present in other species. And starting from the left, I don't know if you can read, we have 2 melanogasters, and the one with the short red line is one that is lacking spawing. If you still see some present, it's because this is basically repeat masker output that takes in account the length and the similarity. So probably this is the most similar Ts that you will find in the genome when you don't have spawing. And the one that is up to 1 is one that shows an insertion that's identical to spawing. And we also saw that it was present in simulants, but it was absent in older strains. And in this talk, I will focus on melanogaster, but basically the story I'm telling you for melanogaster, as far as we are concerned, is probably true for other Drosophilids. The problem is that in no other species we have the same amount of data that we have in melanogaster. And we also find it in Seychellia, which is the sister species, which is endemic to an island. And I will not go into the details of this work, but we got it recently published. Basically, what happened is that the donor species, which is one of the species you see in blue, that has just a few, that probably, I mean, everything I will say as suffers from a problem of sampling bias. There could be a species that is the actual donor, but let's say our number one candidate is a species from the Willistoni group. When Drosophila melanogaster colonized South America, got different Ts from Willistoni at different time. The first one was the P element, but later it got spoink. For Drosophila simulans was the opposite. Got spoink first and the P element more recently, and there's a work from Robert and Christian on that. And basically what happened is that then this T spreads on the entire world population now through vertical inheritance. And the 2 endemic species from the Seychelles, so Drosophila seychellia, and from the Mauritius, Drosophila mauritiana, probably can still hybridize with simulants. So it was a bit easier than to then get the transposon, in this case, POINC. And what happened basically was this transfer, this horizontal transfer in simulants and melanogaster independently, and then simulants deliver it to Drosophila mauritiana and Drosophila seychellia. So yeah, a kind of interesting pattern. How did we figure out? This is our best guess because of all the Drosophilids available in all the available Drosophilids genomes that we have. These are the 250 and more than 1,000 insect genome. The best hit we have is from Drosophila willistoni, but yes, could be that there's another species that was not sequenced that is actually the donor species. Yeah, the best hit for the spawning sequence, yes. So yes, we maybe not Drosophila willistoni per se, but at least a species of the willistoni group is the best candidate. And what we wanted to do next is to be a bit less dependent on luck, we might say, because for the first tease was easier because we had the current genomes and we look at the old ones and we compare the difference, and we wanted to generalize this idea. And so Ricardo, and also with the help of Anna, designed this great software called Genome Delta that does exactly this. So what we did a bit manually and in an ugly way in the past now can be automated. And the idea is the following. So if at the moment there are 120 transposable element families, each one with multiple insertion in the genome of melanogaster, if I take this genome and I map reads that are coming from a genome that has, for example, just 115 of these transposable element families, there will be some gap in the genome because the short reads don't have this information, don't have these transposable elements. And so what the gap is telling us is that if I compare the recent with the old, there is a difference, and these different T's are good candidates that then we can validate for horizontal transfer. So an example, here there's Suslik, but you can think for the P element, if I collect a fly now or in 2008, and I map reads coming from Canton S from 1935, all the spots in the genome that that contain the P element will have no reads mapping. And basically what the software does is looking for the spots and then give them a score, and then there's a bit of manual work validating to making sure it's true. But basically this is what we did, and we cannot do this for the first three T's that I show you because the samples from the museum only provide short reads. We don't have long reads. So the first analysis I show you is long reads, but don't worry, it still works. What we did was applying genome delta, and what we found were 11 T families. I think we use the most recent assembly, which is from 2016. We actually used multiple just to make sure. But basically, we got these families. And as you can see, there are the 4 known one, which is already good. It means is a sort of control that the software work. And then we found Spoink, which was also good. But then we found 3 more. MLE, that now it's been renamed MacLe after suggestion of our reviewer, Suslik, and Transib1. And so if I check from a long read assembly of Canton S, in this case, so a strain collected in 1935 that was maintained in the lab, and I look for these sequences using, in this case, repeat masker, I don't find them. I don't find any of them. And the 3 here, you can see them as a control, the 3 present, because if you remember, they invaded in 1875, probably, or something like that. So here are present, and also all the other T families of the melanogaster are present, but these 8 are missing. And these 4 are also our sort of control, also Spoink now. So these 5, we expect to not see them. But if we look at a recent assembly, we should find them, and we should also find the other 3. And this is exactly the case. And I will also use this, oh no, I will also use this figure as an example to tell you basically what are these transposable elements, because you see them just as basically lines on a screen, but each one has a story. And I think the pattern here is kind of nice. So these 2 guys, Hobo and the P element, are DNA transposable elements, DNA transposons, and usually they have internal deletion and they are shorter. And this is exactly what we see in this assembly. Also, transib1 is a DNA transposon, but so far we have not seen the internal deletion. Let's see if we will see them in the future. Then we have the I element. Also here you can appreciate, if you are like T aficionados, a pattern that is common for non-LTR retrotransposons. And where there is the 3' reverse transduction that leads to truncations, which is the case here. And all the others are LTR transposable elements. So they have an LTR, a gag pole, and in the case of Tyrant, and also an envelope. And yeah, so basically the story started to change a bit. So horizontal transfers don't seem to be so rare. And now we can check using leveraging on all the short read data we have. So these are, I think, 800 or 600 strains collected over the past 200 years. And basically, this is a timeline of the invasion. I could not fit it all, but it starts from the left. And if you see red, the the transposon is present. If you see green, is absent. So they all start in green, and then at a certain point you start to see red, and then it's only red from them, because usually when they invade, as far as we know, they colonize the entirety of the species, and then they are just vertically inherited. So you will see them in all the following generations. And we go up to the most recent one, which is Transib one. And as you can see, we just start to see him around 2014, 2015. So this is the most recent invader, at least the one I can tell you about today, because maybe Ricardo has some surprises for you in the future, but no spoilers. And this is depends on the assembly, depends a bit how stringent you are with repeat masker, but I will generally say that these teas in total, they make almost 1% of the genome of Drosophila melanogaster. It's around 0.8%, which is a lot if you think that this is totally new. So it's like new material that arrived and invaded the genome of melanogaster in the past 200 years, and it's a bit faster than at least my classical idea of molecular evolution, that it's a bit slower. And we can speculate a bit how the balance is maintained between new T invading and removal. There are some papers suggesting that there is a slight bias in deletion in in melanogaster, and this maybe might explain how it managed to maintain the genome of a certain size. But for the moment, it's speculative. Yes, so what we are looking at in each panel are the same line, but each one is telling us about a different transposable element. And on the y-axis, you have the copy number estimate, and on the x-axis is the time. So basically, for each dot, you will have the same x, but the y will change depending on the transposon you're interrogating. So, like here, a line from 19, let's say Canton S was collected in 1935, and you see it in all the panels, but in this panel it's green because there are 0 copies of Transib 1, while if I look at 412, it will be red with 30 copies because this transposon is present with this amount of copies. I don't know. Yes, yes, this is true. Yes, yes. Yes, yes, but I will say yes, and obviously the more recent we get, the more sure we are, because they also come from different geographic location. Like the most recent one we have are from all around the world, so absolutely. But I think for these 5, we are very sure the scenario you're presenting is possible, but it's, I mean, we will never be able to answer because I will never sequence all the flies in the world. So you could always argue that was there for a while and then took a bit to invade. So was maybe in a population for a century and then it spread all around the world. But if we assume it a bit as a Palmitic population, I would expect them to all have it in few years. And maybe it's getting faster now, but I will now show you maybe why I think so. So this is the most recent what Transib 1, and here I'm showing you some of the line collected before 2013. So it's absent. There are actually remnants of an old invasion that look like Transib 1, but there were no full-length copies. They were maximum like 30% length. And here I'm showing just Europe and the US, but we also have parts of the rest of the world, but it was a bit inconsistent, so I just showed these two. And then in 2014, we start to seeing it appear in US and Europe. This doesn't mean that the invasion started there, could have started somewhere else that's not in the data. And then by 2015, it's almost everywhere. And then for every basically strain we collected after 2016, Transib is present. So in, I would say, like 3 or 5 years, is basically it basically invaded the entire world population. So also, I mean, maybe we cannot generalize that, and maybe nowadays getting faster because there's more transfer of goods and people around the world, so maybe also flies mobilize more and it's a bit faster. But if this pattern is kind of shared by the other T, I would be pretty confident that everything like after the 50s will still, like if we see it absent, it means it absent because in few years we would expect it to see all around the world. But yes, we will never be 100% sure. And I will just conclude, since our story with transposable elements in Drosophila melanogaster started with this Kidwell experiment where they did hybrid dysgenesis and they look at the gonads, and the gonads were basically absent in one of the crosses. We thought that maybe it could be interesting to see if this is not just a funny example, but can also be shown for other Ts. In literature, Hobo is supposed to do it, but Robert can tell you better than me that it's actually difficult to obtain this phenotype. And for the I element, there is a different phenotype that is present in the F2. But basically what we did was the crossing scheme of the of Kidwell's paper. So you can just look at the left of the figure right now. And we have this strain called AK8018 and COR018. And the difference is all the T's are shared except for transib1. So one strain has transib and the other lacks transib. So if we cross the strain with transib, so the male strain has transib and the female doesn't, we have around like above 95% of the ovaries dysgenic. While if we do the reverse cross, so we have the strain carrying transib as female, we don't see the dysgenesis, which is what we would expect, because if you remember what I mentioned before, the pyrenees are deposited by the mother in the ovary. So this, if the female strain is the one carrying the transposon, is, let's say, protected. And the same is true for 10.015 and Tom.008. These are just two different strains because we wanted to be sure that it was nothing that was maybe related to the genetic background of the strain. But again, the only difference between these two strains, at least transposable elements wise, is the presence or absence of transib. And by now you learn, so what you expect is proportion of dysgenic ovary is 100%, 90%, let's say. It means that the male is carrying transib, and in fact Tom is the one with transib, while in the case in which Tom, in the cross, Tom is the female, we don't see any dysgenesis. For this data, there are like 5 replicates. Each one I check 50 flies, so 100 ovaries, and then I got the proportions. And just to be sure, we did some other crosses, and here I can give you a brief recap. So these are the most recent transposons that we find. All the other are shared by these strains. So DML68 lacks the P element and is used for checking, is used in experiments with the P element. And obviously all the one that invaded after are missing. Harwich has the P element, is missing all the other. While for 10 and Tom, the only difference is 10 is missing transib 1, while for Tom we have them all. Don't worry, you don't have to learn. There will be a recap in every slide. So this is the classic cross that Kidwell did. So you know the drill by now. DML68 and Harwich, and we have total dysgenesis or partial dysgenesis, or sorry, or no dysgenesis. And this was just to check that our strain with the P element actually had the P element and there was nothing funny going on. And then we have this cross with Harwich and 10.015. So why we need this one? Well, because we don't know if Spoink, MacLeay, or Suslik also cause dysgenesis. And what seems the case here, at least in the F1, there's no sign of dysgenesis. So independently of the direction of the cross, there's no dysgenesis. Consider they both have the P element, they lack the LTR transposon. So Spoink, and MacLeay, and Suslik are LTR transposons, and we don't see any phenotype. And finally, we have Harwich with Tom. So Harwich is usually the inducer, is the strain with the P element that cause the dysgenesis when it's crossed with DML68. But in this case, Harwich, if we have like Tom males and Harwich female, we have dysgenesis, which means Tom is the one carrying the transposon that is inducing the dysgenesis. In this case, we think it's transib 1 because the other 3 didn't do anything when they were alone. So probably it's transib 1. And while when we have Tom females, they have all the transposable elements, so they have all the pyranid protection, and we don't see any dysgenesis. And probably you heard about dysgenesis, and just I just show you the cartoon, but this is what this hybrid dysgenesis look like. So I consider it, luckily for me, it's a clear phenotype. So okay, I cannot move the hand, but basically this is a dysgenic ovary, this is a healthy ovary, dysgenic, healthy, and these are two healthy. And yeah, so we think also Transib1 is able to cause dysgenesis. I didn't enter the details here, but also appear to cause dysgenesis at low temperature, while the P element usually causes dysgenesis only at higher temperatures, like above 25, while for Transib we see dysgenesis also at 23, I think, and maybe even 18. So to conclude, just an overview, what I wanted to tell you is that transposable elements are a lot of fun, even though I know most of you will probably just use repeat masker to remove them from your analysis because they're a bit of a pain, but they are very dynamic. They jump within genomes, they jump between species, and they make a mess of phylogeny sometimes, but they are really cool, and I think they play a role in evolution by providing a lot of raw materials, and they also are faster evolving than what we probably expected previously. And this is what we know about Drosophila melanogaster now, 3 years later. So it was not just Tyrant and P element, some weird funny exceptions. It appears to be a thing that is way more common than we previously thought, and that it happens often, and it's also very fast. And I think, I mean, we're not rewriting the history of evolution, but challenges a bit some assumption where horizontal transfer is a thing that happens for bacteria, and antibiotic resistance. We see it also in eukaryotes now, and I hope you will remember this, that almost 1% of the genome of melanogaster is a passenger that just recently arrived, and we don't know how long we'll stay there, but for sure we are curious to discover it. So that's all. These are the 11 T invasion we uncovered, and that's all for today. Thank you to my amazing supervisor and the great members of my group, Matthew, Ricardo, and also Anna, Misa, and the other that are not here, Siraj, and all the people of PopGen. It was a lot of fun. I learned a lot during these years, and thank you so much for being here.